My lab's really interested in understanding the kinetics of the T cell response. And what I mean by kinetics is when I referred to earlier about the speed of granule, or the velocity of granule recruitment to the membrane, how long it takes for all of that machinery to be inserted into the membrane and granzymes to enter the target cell, and then how long the T cells take to detach. So quantitating all of those steps along the way, if you like, is understanding the kinetics of the response. And so we've done this very carefully, where we've compared, we have a publication where we've compared TCR, or T cell receptor mediated interactions, versus CAR interactions in the same cell. So we have a dual specific T cell, so the T cells express the CAR and the TCR, which enables us to really finely study the kinetics and comparing those receptor responses in the same cell. And so when we did this and we looked at their ability to serial kill, we actually found that it was the same. So we found that the CAR T cells were just as capable at serially killing their targets, or sequentially inducing cell death of tumour cells, just as efficiently as T cells. The reasons why we might want to understand how T cell receptor signalling differs from CAR T cell signalling is that when we're designing treatments and cancer immunotherapies, we may not be using these CAR T cells in isolation. We may want to combine them with other drugs, inhibitors that target various pathways. So if we can dissect out the different signalling pathways and really deeply understand the differences in the kinetics and the cell fate to the T cell of signalling through one receptor versus the other, it gives us all the information we need to design better combination therapies going into the clinic.